And now the last speaker for this morning session is Anton Ussi from EATRIS, based in Amsterdam, and he will give a talk on the quality and reproducibility of data at preclinical level for the development of personalized medicine, a multi-omic perspective. Thank you, Anton.
Thank you, Jacques. It's a very long title. So my name is Anton Ussi. I'm from the coordination office in Amsterdam of EATRIS, which is the European infrastructure for translational medicine. Today I want to talk about quality of data, because eventually anything that you want to get into a patient has to go through quite a severe regulatory process. And so quality of your data package is of paramount importance. So just a quick slide on what EATRIS is. EATRIS is a sister organization of ECRAN and BBMRI. We work together, for instance, in the Corbel project that was mentioned yesterday. And we provide access to about 108 research facilities around Europe for people who want to validate their biomarker or develop a drug or do a first-in-man study or do molecular imaging for drug development. It's very much about helping a researcher take their discovery and try and bring it towards the patient. So we have a distributed network in 13 countries, and we have really a focus on the patient and patient-centric research and trying to help understand the steps necessary to go from where you are now to get towards the patient, including understanding the regulatory pathway. So personalized medicine works. Stratified medicine is already very much well utilized in the clinic. We see that stratification of patients increases the likelihood of actually passing milestones in drug development. And basically nowadays, I think very few medicines are not really personalized in some way, stratified or targeted to a specific patient population. So we see that personalized medicine has started. And as the technologies advance, we certainly are getting much more complex approaches to this. So the question is, how do we make sure that we can continue this revolution and continue towards a new molecular taxonomy of medicine and try and understand that? So personalized medicine really does have a lot of potential to improve the clinical environment. And if we can fix the pricing problems of some of these more expensive drugs, it's obviously a major revolution in healthcare. So we can improve survival, we can reduce wasted effort. I think depending on which indication, for every 10 drugs you give to a patient, for instance, in cancer, only about 2 or 3 of those patients will actually have a benefit of it. This is pre-personalized medicine. So on aggregate, it's actually only about 45% of any drugs given will actually have a beneficial effect, but obviously 100% will have the side effects. So moving towards stratifying and understanding a patient's molecular profile is obviously a very important development. So personalized medicine is here, it's here to stay, and it can also improve the drug development process as well to try and reduce the timelines, help people fail bad drugs faster. So it's certainly somewhere we need to be working on. But beyond genomics, we got a lot of interesting developments around the technologies to help us understand the molecular cascade, to go beyond the genome and try and understand how epigenetics, the microbiome, and how environmental factors can influence our health. So we have a lot of fancy and wonderful new technologies, and it's excellent to be able to make use of these so that we can really go into a multi-omic approach. But it is a complex endeavor, and we have to make sure that we understand the context of use anytime we're using new technologies. A big challenge is understanding what is that technology telling us and what is it not telling us. And understanding early on what are the sources of variability that are being brought into the research process, either inherently through the machine or in your research process. So in order to be able to enable this kind of complex research with using multivariate analytes, you need really reliable data, and from lots of different disciplines. So it's a very multidisciplinary approach, requiring very, very high quality lab and post-lab processes. And with any luck, at some point, we're going to have each of us having a multi-omic personal profile where we're our own benchmark, we're our own baseline, and through a panoply of different markers and biomarkers, we can understand our own health situation and compare it to perhaps to maybe a pool of people like ourselves, based on the same multi-omic characteristics. This is far in the future because we have a lot of work to do before we can have reliable and complex enough data sets to be able to facilitate this. But already today, for each of the single technologies, each of the single analytes, each of the lab processes we do, we have to keep an eye on the quality in order to make sure that we're actually building a reliable record. So that the biomarkers that come out, the biomarkers of relevance can be validated, that we can have strong, robust evidence that the biomarker does play a role in the pathway that we're looking at, so that we can understand the effects of that, so that we can really go towards 4P medicine, which is also preventive to help us understand people at risk, which is predictive, and also which is participatory as well. Yesterday, a lot of discussion. The one colleague from Brazil and Queen Mary's talking about GDPR and data privacy. We're not going to fix that quickly, but the patient owns their data, and so participatory is also about empowering the patient to own their data and say, I don't want GDPR. I want you to work on my data. I want you to use that. So there's also a lot of non-academic, non-scientific elements that we really need to think about as researchers and see how we can enable people and also enable patients to get around some of the things we can't get around, especially GDPR. So biomarkers are an essential part of personalized medicine. And even when you're looking at artificial intelligence and machine learning for decision-making, that's based on the literature. It's based on evidence generated through understanding what a biomarker does and trying to be able to do a little bit of a black box approach to see how all of those different systems interact, to try and get an understanding of what's happening with the individual patient. But the problem is we have an enormous number of biomarker publications each year, 40, 50,000, something like that. And how many of those are actually taken forward towards clinical validation? Very, very few. It's because it's very expensive. It's time-consuming. The data that you have to present, if you would like to actually get this into the clinic, is very high. There's also a lot of danger associated. Even if you want to use a biomarker just for your drug decision-making processes, say a pharmacodynamic biomarker, to be able to say yes or no to a drug in development. If you're not 100% sure that biomarker is telling you what you want to know, you could be killing a $2 billion a year drug because you're basing it on a readout of a biomarker you can't rely on. So the provenance, the quality of the data that you're generating is of primary importance at any point in the process. And trying to get through this run with one of those 40,000 biomarkers each year is very challenging. Even the pharma industry has a really hard time with it. So the biomarker development pipeline, basically from here going through here. So your first retrospective validation of the analytical method and of the biomarker itself and of the process, developing an assay, and then trying to do a prospective clinical validation. There's fraught with, well, firstly, there's a huge drop from that first discovery, so you got your publication, an enormous drop to the next that'll actually start in that next process. And there's a lot of weaknesses all along the road. Reproducibility of the primary data is one big one. So the reproducibility crisis, I'm sure you're all aware of the issue around reproducibility. I'll come back to that in a moment. Access to the right quality biosamples, making sure that they're actually fit to task, making sure that they've been qualified before you use them. If they've been stored well, I'll come back to the pre-analytics in a moment as well. The verification of data. If you want to access large pools of data, we simply just can't nowadays. And so even if you can get your hands on it, is it interoperable? Is it reliable? Is it accessible? And coming later in the pipeline, collaboration with industry is challenging, but it's also essential, because even diagnostics compared to drugs development is still relatively cheap, but relatively cheap means tens of millions instead of hundreds of millions. So collaboration with industry is essential. And there's always market uncertainty. So I saw one of those diagnostic kits. The photograph here was the one kit from the Netherlands, and it took them many years. And I had a conversation with the CEO at the time when they had beautiful data, amazing data sets. They had the CE marking for the diagnostic, but they couldn't get reimbursed because it was quite early on for that type of diagnostic kit, and they couldn't get reimbursed because the healthcare system didn't understand what to do with it or how to do it. How was this going to affect them? They were afraid that they were going to start spending too much money on their healthcare process. So even when you get here, you think, great, I've actually done this, the one in 100,000, and then nobody wants to give you money for it, even though you're damn sure that you're actually going to help the healthcare system. So there's a huge, complex array of difficulties along this road. I mean, as academics, we're mostly here. So today I'll focus just on a couple of issues around here. One being the reproducibility of the data. It's a crisis. It's a real crisis. And slowly we're starting to look at that and see what we can do about it, but we've got to move a little bit faster, to be honest, I think. So each one of these is a study in ALS. Each one is a study showing the effect size of on animal tests, to show the results. This is the effect size published in the papers. And then an independent lab went and actually tried to repeat the results, and these were the effect sizes they got. It's quite terrifying results. And I think this is probably going to be the case of any indication area. Because there's a lot of types of sources, there's a lot of sources of variance in the lab process. And those sources of variance can come just simply from the lack of quality systems, traceability of your processes and your SOPs, going down to troubles having repeatability. Maybe the temperature of the room at the time you performed your assay can create a different outcome. Cross-lab replication, even if you do that, which it really comes late in the stage for most academics or never at all, is very challenging as well. And obviously then going across platforms as well, and then to try and generalize into a population, well, that's the cherry on the cake. So these were 5 studies where companies tried to replicate key findings from high quality and low quality journals. So Amgen, they replicated 53 seminal studies, and these were big studies, Nature papers and those sort of things. And these were basically, this is the non-reproducibility rate, not the reproducibility rate. So Amgen, they must be a really terrible company because they only managed to replicate 11%, or there's an issue with the data. So probably the latter, but you can see that we have a real crisis. Nobody was able to replicate more than 50%. So it's estimated 50 to 90% non-replicability, non-reproducibility in preclinical research. And the problem is, you guys, so most of the talks we've been hearing today are about artificial intelligence, machine learning. What do they learn on? These data sets. So what are they learning? So we have a long way to go. And so while we work on the AI revolution, you have to be careful. What's the provenance of your data? What are you actually looking at? Because there's going to be a lot of confounders in there. And there's a lot of causes of reproducibility. So Leonard Friedman, this guy's been doing amazing work in the US. He was with the Global Biological Standards Institute looking at the source of variability. Look at his papers. It's just a few papers, but look at his papers. Very interesting and very helpful because you can see there's a lot of actionable elements of the research process that you can look at and fix without having to change anything. So things like incomplete reporting. So this in itself, incomplete reporting, doesn't mean that the study is wrong. So that's not necessarily a source of variability. But a lot of these are. And so lack of validation of reagents is a major one. One of our institutes in Czech Republic did a protein analysis of the NCI60 cancer cell lines, and at least 4 of the cell lines in there were not the cells that were supposed to be. And these are the NCI60. Imagine how many people are using these cell lines. So you see that there's, in the lab process, even accidental, but then there's also accidental or non-accidental biases that come in. We heard a good comment earlier saying that the method chosen was the perfect method for that model. Yes, exactly. So we're also going to be doing a bit of the p-hacking. So there's enormous sources of variability, and they're coming before you generate the data. Once you have the data, it's already too late. Because if you look here, 75%, so 36, 27, 10%. These are the sources of variability. Reagents, reference materials, more than a third. Study design, laboratory protocols. All of these are immediately actionable now. And they have to be actioned before you apply for your grant, because you have to design the study to get the grant, but design it well already. And in the lab process itself. So variability obviously comes in many shapes and sizes. Biology is complex. We heard that before. Yeah, it's biology. So the heterogeneity is a major issue. But also the processes. There's a lot we can do in terms of the processes. So I'd like to just spend a moment also looking at the process, because the last talk from Elmer, all your work is being done on cell lines. Right, so it's so important how much of the research work, the preclinical research work, is being done on cell lines or on patient-derived tissues. So you have to understand that there's a huge amount of variability that you can bring into the process if you're not careful with the pre-analytical process. So pre-analytics, it's basically the moment from you've drawn the blood or you've taken your biopsy all the way up to the moment that you analyze it. And there's a lot of things can go wrong there. And also, if you think about it, if you've developed a biomarker that's great in your lab, is it going to stand up to the fact that maybe somebody is driving around the countryside in France with the sample in the back of the van for 6 hours before it's getting to the hospital, before it's being put into the freezer or aliquoted? So there's a lot of things that can go wrong in this process, and it's very important to be sure that when you're getting samples or when you're sampling, to try and control the pre-analytical processes. Here you see the freeze-thaw cycles, and it's quite a significant effect on the cytokines of in serum and plasma if there's been a few freeze-thaw cycles as well. So you've got buy something from the biobank. It's a very high-quality biobank. So these are qualified samples, but look at just a few freeze-thaw cycles, and already we have possibly basically useless samples. Room temperature, the effect on IL-8 it speaks for itself, really. So when you're doing your research at the design phase, I think both as a research funder, because you're the gatekeeper, and people do what you inspect, not what you expect. So as a funder, you have to keep your eyes on everything. But also as the researcher, make sure that you go into biobanks that are accredited or certified, that they have strong processes, and that they're giving you a report on the provenance of the samples they've actually also qualified the samples with what readouts, and so that you really can have confidence what you look at. It's probably more expensive than the samples in the fridge at the laboratory next door from your friend. But if you want the research to be reproducible, it's an important step to take, is to make sure the samples are very high quality, and they have to be fit for purpose as well. You have to bear in mind for some analytical processes, for some readouts, a specific sample type might not be a suitable sample type. So you also have to talk to the technologists, the biologists who understand the use of that specific analytical technology. So when you're designing your study, work with the technologists, work with the biologists, work with the clinicians, make sure there's multidisciplinarity in your design. Otherwise, you are assuming a lot of things. And you're very smart people, but you probably don't know everything. So biomarker development, which basically is the basis of any personalized intervention we could wish, is very challenging. There isn't a single integrated pipeline. There's not a single place you can go where you can go to the biomarker shop and they're going to help you take it from here to clinical validation. It's getting your hands on infrastructure, facilities, the technologies you need to handle it well are difficult. And your job as an academic is to make publications, which is to make discoveries, not to validate. It's boring. I'm not going to get a Nature paper by validating something. So the confirmatory part of development, the second you've got your publication, you move on. Somebody has to take that over. The research funding organizations are now funding translation a lot more. They're also starting to fund replication studies, so that's a good thing. But we need to also find a way as a system, as a research system, to reward and to give merit to people that are doing collaborative research and that are doing the less sexy type of research around confirmation. Reproducibility is a big issue, and the biomarker variability does not help it whatsoever. So just 2 last slides. There are things we can do, I think, from today. So EATRIS, we've been running since 2014, and what we see is that just by looking at the funding process and the research process, we have a lot of opportunity to help good research plans be written before the actual funding happens. So if you think of the classical funding process, a call opens, right? Might be your National Research Council, and you say, that's great, I'm going to apply for that, and I want to validate my biomarker and get it towards the first prospective clinical validation, and I write my plan to do that retrospective validation. What you're doing is you're a diagnostics company. You're being asked to be a diagnostics company and write a preclinical development plan for a product. Because the moment you're doing translational research, you are developing a product of sorts, because your intention is to get to the patient. And to get to the patient, it has to be something identifiable that a regulator can say, yes, I know what this is, and this is good. That the healthcare system says, oh, this is good for my budgets, and the clinician says, this is good for my patients. But you're an academic, and you're told, make an application, 70 pages, and we'll tell you yes or no, and if we give you the money, then you can't change your study plan whatsoever. So you're told to be a diagnostics company, you're not given any money to support in the grant writing process, right? So you can't get a regulatory consultant on board because they cost you 50,000 euros to write a preclinical development plan. So you're expected to be able to know how to do everything. And then you get the money if you're lucky. And that review process as well is a bit focused on scientific discoveries. So also inherent methodological weaknesses in the study plan aren't easily identified because the review process is being conducted by clinicians and academics, not methodologists, not scale-up experts, not engineers, not assay development experts. So there's a lot of gaps in the actual process to where great science is being funded, but great science doesn't get to patients. It has to be feasible, and there's an element of non-scientific review that needs to be included in that. And then once you get the money, it's really hard to change scope. Suddenly, you're about to start your project and you hear from an expert, that sample type is wrong. You're going to have to change that sample type, but it happens to be a different biobank. Then making those kind of changes can also be very, very difficult. So as a researcher, as a research funder and as a researcher, you need to kind of work together. And it sounds a bit weird. The research funder doesn't want that because there's conflict of interest, right? There's supposed to be this arm's length process. Peer review, no conflict of interest. But at the same time, the research funder is an investor in research outcomes, not just making sure that you don't get in trouble for your portfolio. And so to be an investor in research outcomes, you need to work with the researcher, encourage them to access the sort of expertises they need to be able to do good study design. So think about a two-phase selection process. Phase 1, the researcher comes just with a scientific concept. I think this biomarker is going to help me be able to understand the difference between latent and aggressive tumor type X. This is a clinical case. This is what I want to do, but not a study plan, just saying I want to get to retrospective validation. And then you do your classical review, you have your clinicians and you have your scientists look at it and say, yes, the science is good, the clinical case is good. And then you invite them for phase 2, thinking, if you come back with a study plan that reflects this and is reasonable, then we will fund you. And so maybe that's 20% of the proposals. But then the investigator knows they have a good chance of getting funded. So then they can go and invest the time and the money to actually get the technologists and the people on board to design the study well. So by phasing it this way, you have a better chance to get the really rigorous study design on board. But then also as a funder, you need to access these sort of technologists and these sort of people yourself to review. You need people who have experienced the development process to look for the methodological weaknesses. So it does have increased burden on your review process, but it's a necessary part of the process if you want to have the maximum chance of the research getting to the patient. You need to really look, is the plan patient focused? We perform feasibility assessments for funders sometimes. they ask us to review the non-scientific elements. And a lot of times you see that the project isn't patient-focused, it's publication-focused. So it's got the name translational on it, but it's not patient-focused. So it's really important to bear in mind, who are you doing this for? Why are you doing this? It's in order to make people better. And so you have to look at the patient, identify the patient on whom this thing is going to work, and then go backwards from there, reverse plan from there, and take the steps necessary to get towards the patient. And as a funder, it's important to be able to look at these things as well. I think that's it. Really important to bear in mind that all of the data we're generating is feeding our training sets. So in law, for commerce, the term caveat emptor, buyer beware. So when you're getting data sets, be careful, beware of the reproducibility of the data sets and what you're working on. And I think this is a systemic problem that we all need to work on together, but it's really important if we're going to actually have a chance to improve the process of finding the drivers of human health and human disease. Thanks a lot. Thank you.
Thank you, Anton. Some question from the floor? Yep. Thanks.
Thank you, Anton. When we analyze transcriptomic data, it's very easy for us to see who is male and who is female. We notice that there is a lot of mislabeling in very reliable and famous labs. So, like, although people do take care of quality issues, it's still things that are human error. Like if they change the gender, they may change everything else. So what I'm wondering is that is there a guideline, specific guideline by EATRIS that you can follow to make sure that all these issues that you raise is followed? Like PRISMA for meta-analysis, and several other guidelines for each some of this.
I don't think there's one specific volume somewhere, but there's lots of different bits here and there. So we're involved in a large rare disease initiative called the European Joint Programme in Rare Diseases, and there we're putting together actually a portfolio of resources to help people do translation. That will come on the air in the next few months, and it'll be a living resource where more and more things will be added. There will be things like so NIH NCATS has for screening systems developed an assay guidance manual. It's a 300-page volume on developing screening assays. So there's a lot of things out there. There's not one volume. For good biomarker practice, there's quite a lot of publications out there. If you give me your card afterwards, we'll send some. Some of our institutes are quite active in good biomarker practices and espousing how to do that. But I wouldn't say that there's one single volume that tells you how to do it all. But I'll certainly be happy to forward you what I can. Thank you.
Very good talk. I think that everything that you covered is real. That all this challenging, all these problems, and the people, in my opinion, are like, I don't want to see. In your experience, what is the proportion that was the feasibility today for personalized medicine?
The proportion of feasibility of...
What is the impact that today is, for me, in my opinion, is very low because there are many low quality.
Yeah, exactly.
When you did the last one that you put the cross population, is a big issue.
Very big.
And I don't know, it's like personalized medicine. For me, you come, but it's not soon. Maybe it will be 2, 3, or 10 years old. I don't know, because the variability is in all these stages. And the people in the area, in the research, are doing one part. Now our lab are working with omics. And before going that, a lot of different experts in the area, and the one is not talking clearly with the other. This is in our lab. Is that means your experience, how the advice for us that you are starting this?
To be honest, I don't think anybody solved that. The fragmentation, silos, people not talking to each other, people not working together. The whole system is built to be fragmented. The whole system is built on, especially in academia, it's built on a new discovery. It's an industry where we're just supposed to churn out paper after paper after paper, and your whole career is built on that. And so the system itself is not built to get to the patient. So I could imagine, I mean, this is my guess, but I could imagine if we would really look at this, that we could change overnight from now to the ideal situation, we could probably be 20, 30 times more productive, because we would spend a little bit more money on each study. Make sure it's the right study, make sure it's methodologically sound and significant, make sure we look at sources of variability, we plan replication, independent replication into the study. It costs more for that one study, but I'd rather one of those studies than 3 cheaper studies that we can't really rely on the provenance of. But the whole system is built to be fragmented like this. So it is important as a system to start looking at ways of defragmenting and coordinating things and looking at the systemic problems. We keep looking at the projects. Let's fund oncology. Let's fund ALS. Let's fund this. But we don't look at the box, the system itself. And the system itself is not working very well. So as research funders, as directors of research institutes, look at the process. And if you can fix the process, you're going to have higher quality research coming out of it.
Thank you.
Thanks.
Yeah, very nice talk. And I have experience in participating or advising groups in experimental design and the importance of quality control. And yesterday we were talking about harmonization of the data, the importance of quality of the data, and everywhere I go and I hear some people like you and you see the audience, everybody is acknowledging you and moving back and forth the head. So everybody seems to understand the importance of that.
Yeah.
Everywhere you go and everywhere I go, I saw the same reaction. But then it seemed that after the talk, everybody forgot about it. Yeah. So what do you think nobody applies that, knowing that this is a key point, because everybody wants to avoid garbage in, garbage out.
Yeah. Yeah.
This is the first question. And the second question is, yeah, right. I mean, everybody is arranged for the publication. But what do you think, or what is your suggestion in order to move away from the publish and perish dilemma?
So for the first question, I think you're under a huge amount of pressure to write a grant, perform the research, get published, right? So you do what's necessary to get the grant and to get published. So if you are writing a research proposal that's maybe not so methodologically robust, but you know you'll get a grant, or that's not going to be the reason you don't get the grant, and you know you're going to be able to get published with 6 animals instead of 12, then why would you do 12 animals? Because you're not going to write a grant that looks expensive, because you know everybody else is writing the grant with 6 animals, and they're promising to solve Alzheimer's with 6 mice, not 12. And so this, again, the system encourages you to do the minimum necessary, not the maximum necessary. So the system, that's why I said before, people do what you inspect, not what you expect. So it's up to the publishers and it's up to the funders to really have a rigorous look at the study design, the study plan, really be a co-investor in the research. Spend more on fewer projects. And I forgot the second question already, I'm sorry.
How to move away from the publish and perish?
Well, I don't think we'll move away from it, and we don't have to. I mean, it's academia's job to make the new discoveries. We don't want you to become drug companies, but I think the system itself should be able to, firstly, improve the quality of the publications through that spending more on less, but then creating the system where the system picks up good science and automatically takes it along. So everybody should be doing what they do best. So you should have a technology transfer office that has the expertise to help you be able to file a patent on a molecular signature. You should have organizations that can help you get the follow-on funding to actually then write a good translational plan, try and get it to a point where it might be interesting then to license out to a company. So it's just about putting together the conditions around you with people that know what they're talking about, people that can help you write a translational study plan if that's what you want to do. But if not, at least you have rigorous research that could you potentially collaborate with a company to develop further, these kind of things. So you shouldn't be expected to do translation if you don't want to do it. As an academic, you should do discoveries. But no good ideas should be left behind. Every good idea should have the chance to get to the patient. And we have to build the framework for that. And it's very complex, but slowly it's coming into place. EATRIS was created to create this ecosystem where you can come to us and say, okay, I want to do a retrospective validation, and we help find the biobank that can have the right samples and do the qualification, the analytical readouts, the technology centers that have the best technologies. So it's starting to happen slowly, but it's a very, very long process.
I want to comment on this a little bit further. For example, recently in European funding studies, it's required to produce a DMP, data management plan. Yeah. And I think this is a great idea because it kind of gives the idea that the data belongs to the community. Yeah. However, I've seen a lot of cases where researchers are unprepared to write a plan. The offices are not coordinated or prepared. And down below, the researcher knows that nobody's gonna not fund this research because of a good data management plan. So I think it's a good idea, but it's not yet what we need to make the best of our data and to bring on board the, competencies that we need. Yeah. So again, I completely agree that research should not be, a research funding proposal should not be assessed only on the basis of the science, but also of many other things that benefit the community. Yeah.
Yeah, absolutely. As you say, the data management plan is a very good step. But yes, a lot of people, it's window dressing and they don't really care. And even if they do put it on some database somewhere, who's going to find it? Is it interoperable with other data sets, et cetera, et cetera, et cetera. But at least it's a start. And I think we just have to keep looking at the process and keep asking ourselves, is this fit to task? Can we make the process better? And if we can just keep iteratively improving on the process, not just the science, then we can have a lot more efficient use of the funds.
Thank you for your elegant presentation. Only a comment. In your point of view, when is the best time to introduce industry or pharma company in the cycle of the research and translational research? Yeah.
Unfortunately, it's very late. It's normally about 5 years and a few 100,000 later than you can do it, because of the reproducibility problem. A company will not look at your data until you have some really nice independent verification. Let's say you have a new molecule for a drug. An adventurous venture capital will only look at it if you have a target that's been very well validated in the literature. You have a really good signal in at least 2 disease models, a signal of efficacy. There's no easy answer to that, but you basically imagine there is no answer here. But basically, you've got to build very convincing data, very convincing data. That's a lot of work beyond that first publication. Several years and several 100,000 euros basically will be the minimum, even for a biomarker, I would say. There was.
I think everybody was impressed when you showed the reproducibility data, but you also put the names of the companies there. Yes. So if I were in one of those projects, I would feel embarrassed. So, no, but it's true.
Those companies, they were the ones trying to replicate the seminal findings. I'm assuming that the companies did a pretty good job at trying to reproduce.
So the thing is, when you show that and you are able to analyze some data and again trying to show this, it's not a way of, at least if I publish a paper, I know it's going to be analyzed in this sense. So publishers start doing this kind of analysis and grants. People who are evaluating, they know that if they gave a grant and then the reproducibility is not good, and you can show that. Like, it's a way of changing the thing.
Yeah, like the system has memory. Like the funder will remember you, like because the last grant they gave ended up not being right. I don't think the system has that much memory. unfortunately. If a funder would be able to look back and say, yeah, they weren't able to replicate your findings, we don't think you're doing good science, that would be obviously really important, but I'm not sure that the system has that good a memory, unfortunately.
It's good in itself, no? Because I mean, research is a risky task. Yeah. So if funders only fund what it works, it's not research anymore. Yeah. So I don't know.
Yeah, no, true. But I think you want to, but you still want. It is risky, but you want to be able to believe the result, even if the result is negative. Yeah, yeah.
Yeah, I have just a couple of examples about this aspect because, for instance, in the field of radiomics, so the analysis of the imaging, it's like 5 and maybe 10 years that there are a lot of publications that are mostly garbage. So what the community did was to create a kind of international score, which include also a value for the reproducibility of the data, and also if the study include or not a validation inside.
Nice.
And so they create this list of all the publications giving this score, which is helpful also for who wants to make new studies about that. So because it's like a kind of guideline, which is something also similar to what was done by the tripod for the definition of the internal and external validation in the studies. And also for me it's also important what some kind of journal are trying to do, or at least in my field, because there are, for some specific topic, they ask you the validation, otherwise they don't take into account the review processing. So the editor stop at the first time the evaluation of the paper. I think it's something quite interesting and valuable.
Yeah, exactly. These are very good developments. Yeah, you do see the journal starting to move now, for sure, indeed. Yeah, and the development of the radiomics community, that sounds like a really good initiative. So it's the community taking action itself before they're being forced to by others. I think that's great. I think it's really important. Yeah.
I just wanted to add on the same note. I mean, obviously, it's a whole ecosystem that has to change because the